Hello, this is Neil Levin with Now Foods, and today we're going to be speaking about quality beyond measure. Now is, most importantly, a family-owned business. We're considered the largest family-owned company, the privately owned company in the natural products industry in North America. And we're proud of being family-owned because that allows us to make the right choices at the right times for the right reasons, as our CEO, Jim Emmy, often says. And he's right. It means that we can devote resources to quality and to helping with industry associations, setting up standards for quality that the industry uses, and participating in these processes without regard for how it affects quarterly profits, without regard for stock prices, without regard for trying to have a venture capitalist or outside investors get their return on investment that they would like to get. These are all things that regular companies that have stock prices publicly traded or outside investors have to worry about, where we can decide to purchase buildings, set up extensive laboratory and quality facilities, hire so many quality professionals, and do all the other things that are responsible really for a lot of our reputation. Now is also a company with a mission. Our mission is to provide value in both products and services that empower people to lead healthier lives. So our main manufacturing facility in Illinois opened in 1998 and is over 250,000 square feet. In 2012, we opened a manufacturing facility in Sparks, Nevada, outside of Reno in Northwest Nevada, where we manufacture capsules, soft gels, foods, various other products. And a couple of years ago, we opened a distribution center a few miles away from our main manufacturing facility in Illinois. And this one is located in Roselle, Illinois, a neighboring suburb. It's 225,000 square feet and has the latest technology for efficiencies in distribution. So the products that we distribute to our distribution center in Nevada, we have 3 distribution centers in Canada as well as manufacturing in Ontario. And we also distribute to our partners around the world that distribute our products in foreign countries. And that allows us to ship efficiently and keep our costs down, which is part of the mission, providing value in products and services that empower people to lead healthier lives. What is value? It's having the highest quality for the best price. This is one of the ways we do that. So we manufacture a wide range of products, tablets, capsules, liquids, soft gels, powders, packets in our modern, clean, quality controlled manufacturing facilities. And we do tons of testing. We're doing approximately 16,000 tests per month to assure quality using the latest instruments and equipments and using highly skilled, trained quality professionals, including chemists. And of course, there's a number of quality checks that are imposed upon us as well. We are third-party quality tested, audited, and verified. Underwriters Laboratory, UL, is our third-party certifier for GMP or good manufacturing practices. Quality Assurance International is our organic certifier that does third-party audits and certifying that we are complying with all the regulations to accurately separate and label organic products. NASC is our certifier for our pet supplements, and we have certified gluten-free products for some of our food products as well. And we have a number of products that are non-GMO project verified, as well as others that are non-GMO that the standards are a little different, where some products can't get non-GMO project verified, but we do have documentation that they're non-GMO. And that's in regard to issues like farm level testing versus testing after it's been accumulated by the processors. And we are, as I mentioned earlier, selling to foreign countries, and that's actually our fastest growth right now is internationally. And that's an advantage actually for domestic retailers as well and consumers, because the economies of scale that are presented if we're distributing to upwards of 70 countries around the world means that the same product, as you see on the slide, GLA from Evening Primrose Oil, if we're distributing that in a number of countries, we're making more soft gels, we're buying raw materials, and the cost of quality testing and purchasing and some of the other fixed costs, setting up equipment and cleaning equipment, those are fixed costs that are now distributed and amortized over many more capsules and bottles. So the cost per bottle or per capsule of producing and testing these products actually goes down because of the volume we can increase by doing not only international sales, but also contract manufacturing or private label for other retailers. So this means that we can keep our costs down and offer a better value to everybody. And this is a sampling of some of the brands that we have in the United States. We actually have some additional brands in Canada. We're not going to be talking about it this webinar, but there's some private brands we have that we distribute in Canada as well. And we do offer many products that have trademarked ingredients. And trademarked ingredients are name brand ingredients. This is a sampling. Some of the logos you might see on labels indicating that these are branded ingredients. What is a branded ingredient? It's an ingredient that has a name, it has a trademark, it often has a patent. And a good example might be if you purchase a car and it came with a standard stereo and you wanted to put in a name brand higher end stereo. Maybe you want a Bose or a Kenwood or a Sony or a Pioneer or some of these brands you've probably heard of. You can put those in and you know these components are brand name components from reputable companies with good reputations. And that allows you to upgrade your experience. We're essentially doing the same thing with our supplements by using name brand ingredients. Typically for a name brand ingredients, let's pick an example, ubiquinol, a form of CoQ10 that's offered by Konica, which is the logo on the bottom left corner. Everybody who buys ubiquinol is buying the same material from the same supplier, Konica, produced in Texas, by the way. And this is something that no matter which brand you buy, it's the exact same raw material. Now, there are differences in how we produce our products. For example, we will use MCT oil as the delivery system for ubiquinol. Ubiquinol actually requires all the manufacturers to deliver it in a soluble solution to increase absorption. And it's not the form of the CoQ10 itself that's responsible for the increased bioavailability. It's the required delivery system on all ubiquinol products. But we get to choose the delivery system. We're choosing a natural extraction MCT oil to improve absorption Other companies might use polysorbate 80 or petrochemicals and detergents like that that are not naturally sourced to improve bioavailability, but they're not necessarily natural or as natural as the ones we're using. So we have choices to make. We have to start with the same ingredient and end up with the same absorption, but how we get there depends on the values of that company. Our value is that natural is better. And this is actually a longer list of trademarked ingredients from our catalog. So this gives you an indication that we have a lot of these trademarked ingredients. And trademarked ingredients are really the same raw material no matter which brand of vitamins or supplements you're purchasing from. If you're buying ubiquinol from us or one of our competitors, it's the same material from the same supplier. And to give you an example of how that can affect other things and how the values of the company and the practices of the company can affect what people are actually going to pay for. There's a chart here I'd like to explain. And first of all, if you're buying the same raw material, let's use ubiquinol as an example. If it's from the same supplier, it's roughly the same price. Now, as one of the larger vendors, and remember, economies of scale apply to not only our purchases to make stuff in the United States, but all over the world, as well as our contract manufacturing. So we're actually one of the largest purchasers of ubiquinol in North America, if not the largest. So of course we have perhaps some advantages in purchasing, but let's assume it's roughly equal for this argument. The testing and quality costs. We believe we do more testing than most companies, and I'll explain some of that in a few minutes. But let's give everyone the benefit of the doubt and assume that the quality cost is roughly the same, as well as the cost of the trademark ingredient, that the smaller producers can actually buy it for as much as us who have more leverage. So assuming that those are equal, what are some of the other factors affecting the cost that you as a retailer or consumer would be paying for the same product? One of them is distribution cost. We typically sell direct to retailers. So the retailer is not paying a third-party distributor a markup to get them the product. We do have some distributors which some practitioners or others prefer to use, but of course that affects the margins. And sales rep costs. The extreme example would be multi-level marketing companies where there's a lot of people making markups between the producer of the product and the eventual consumer. So the more people involved as middlemen or middle people in this scenario, the more costs that are going to be there. Let's look at advertising and marketing costs. Now is actually a very lean company. We do not spend a ton on marketing because we believe that quality and word of mouth is the greatest value to the consumer, and that they get the message that we have good quality products at reasonable prices. So we're not a marketing driven company to get sales like some of our biggest competitors are. We don't spend nearly as much on marketing as them, and we don't do much mass marketing because we're not really in the mass market. We're in the natural products channel primarily. Lastly is overhead and debt cost. We're a debt-free company. We own our own buildings. We have no long-term debt. We don't have to pay these interest payments every month for things that we're purchasing. We buy a lot of our warehouse equipment at pharmaceutical auctions. Remember, this is equipment that's stainless steel. It's designed to be easily cleaned and non-reactive. And we have an engineering staff that can retool it. So a lot of our equipment that we're using is very high quality, but we're paying maybe 20 cents on the dollar versus buying it new. And with our engineering staff, it's actually as good or better than new as far as meeting the requirements that we have. So you could see the exact same raw material, assuming they're purchased at the same price and assuming they're tested the same way, even though we probably do more tests than most companies. that there's a difference in the cost of the company getting products to market. There is one other factor, though, that's not on here, and that's the profit margin. How much are we marking it up above our cost? And if you look at publicly traded companies which disclose such figures on their annual reports, we're running on a much lower margin for profits than most of our competitors, and certainly less than our publicly traded competitors. And now originally was an acronym for natural, organic, and wholesome. We believe that natural is better. And some examples from a much longer list on our webpage, by the way, of things that we don't use in now products. We don't use SLS, sodium lauryl sulfate, which is actually a synthetic fatty acid. We do use natural coconut or palm oil derivatives, which have some similar sounding names, but they're actually natural fatty acids. Some of them are full spectrum, let's say coconut oil extracts, that are very, very different from a single synthetic petrochemical fatty acid. Or we don't use polysorbates. I mentioned the ubiquinol as an example before. We don't use PEG, polyethylene glycol. We don't use a whitening agent, actually a white paint pigment, same pigment they use in white paint, titanium dioxide, to mask visual imperfections in tablets and capsules. Because, for example, on a soft gel capsule, we might be blending some materials together, and unlike ubiquinol, they're not soluble, they're not blended into a seamless liquid. They're actually blended where maybe we're putting a powder into an oil or something, and there's swirls and separations. They don't affect the performance of the product, but they're visually jarring perhaps, or maybe someone will question it if they see there's some separation within a capsule. And so we actually put a disclaimer on the label that such separation is normal, and we're not using white paint pigment to make the capsule opaque or the tablet coating opaque. So you can't see there's speckles or swirls or differences from blended ingredients that are not really reacted together. We don't use artificial colors or flavors. We don't use synthetic vitamin E or synthetic forms of amino acids, which are the D or DL forms in our supplements. And we don't use fluoride, parabens. We don't use irradiated ingredients or nanotechnology to produce our ingredients. And of course, natural means non-GMO. We have an extensive non-GMO assurance process where even if something is not going to have the audit trail all the way back to the farm level, we still have assurances from the vendors and from testing raw materials before they're processed and other means to assure that we're producing products that are non-GMO. A number of years ago, our entire food line became non-GMO. All of our essential oils are non-GMO. They've all been submitted to the non-GMO project for verification with appropriate documentation and audits. And all of our natural flavors, for example, we've converted years ago to non-GMO sourced. And what that means is that if there's a lecithin or a vitamin E added as a processing aid, preservative, or other things like that, that might be from soy, or if there's a maltodextrin or starch that might be from corn, that we're making sure that they're going to meet non-GMO project standards for being non-GMO sourced. And all the products that we label as organic are third-party certified by Quality Assurance International to meet U.S. Department of Agriculture Natural Organic Program standards. That requires separation and cleaning of equipment that will avoid cross-contamination. But we're a very strong quality brand. We've got over 150 people working in our quality departments, quality assurance and quality control. And if you add R&D and the science department that reviews these studies and claims and things like that, and labeling folks and all the other people involved directly with quality, we're about 200 people working in our technical department, and really everyone in the company is responsible for quality, all of our 1,500 or so employees. But when you're looking at about 10% of our staff is working on quality, and that's maybe 10 times the size of our marketing department in terms of staff numbers, we really put our money where our mouth is here. And we're talking about tens of millions of dollars in investment in laboratory facilities and instrumentation, some of the most advanced quality testing instruments on earth, which we'll go over briefly. And here's an example of looking at probiotics and looking at the strains or substrains, the specific varieties of probiotics we're using is a good example of some of the ways that we're qualifying raw materials. So first of all, probiotics are microorganisms. They're defined by the World Health Organization as viable, in other words, live, non-pathogenic, safe, microorganisms, which include bacteria, but also include fungi, yeasts, that have some kind of health benefit to the host. That's the official definition of probiotics by the World Health Organization. But when you're looking at each strain, or sometimes they're called substrains, beyond genus and species, in other words, they have specific health benefits, and not one of these species has all the benefits we've heard about or seen in studies for probiotics. You buy one, it doesn't do everything. It does specific things. How do you know that a strain is going to be safe? You have to identify it genetically. You have to screen it for other microorganisms that are undesirable or even toxic, such as pathogens. So to ensure safety, we have a full micro lab. It's third-party certified. There's a process every year where the microbiologists there are certified, and the technicians, to meet full microbiology lab requirements. We typically get a 100% score, by the way. We use not only the normal test tube Petri dish systems, but we use the Solaris system, allowing us in a day or two to get readings of pathogens, yeast, molds, bacterial count, etc., which gives us a quick screening. If we get any hit for pathogens or other contaminants, we then follow up with an investigation and the full test tube Petri dish type testing, which takes 5 to 7 days. It's a longer process. If we were to outsource this, we might be waiting weeks to get results, which affects the just-in-time or efficient ability to get materials in, qualify them, and process them. We're also, at this point, we believe we're still the only manufacturer in North America that has a ribo printer, allowing us to accurately identify the genus, species, and the substrain, or the specific strain of it, that we can differentiate different strains of Lactobacillus acidophilus, different strains of Bifidobacterium lactis. Which strain is it, and what is the evidence that we can claim on it? Are there human clinical studies on it? Are there generic studies on it? What are the benefits we can claim? So this is a way for us to identify that what we're putting in the bottle matches our label claim. And right now, if you don't have this equipment and the training to use it and the standards to match to, which we've invested in all of that, then you have to outsource this or take your supplier's word for it, which is really what most of the companies are doing. And when we're talking about clinically relevant strains, these are some of the strains we're using. One of the questions that comes up is it a human strain? And that's actually somewhat misleading. The lactobacilli bacteria are some of the most widespread bacteria on Earth. They've been found in the soil, they've been found in the water supplies, they've been found in the air, they've been found in the foods. So where they're first identified is how they're labeled. The origin, if it's found in a human, it's called a human strain. Now imagine this, if I'm working in the garden this weekend and get some bacteria from the soil on my mouth, maybe I'm eating a fresh cherry tomato or something, and that bacteria gets in my system and I'm in a study and they do a stool sample and they find this bacteria they've never found before, it will forever be known as a human strain. It was found in a person. That doesn't mean they know what it is. That doesn't mean it's safe. That doesn't mean it has any efficacy. It just means that happens to be accidentally the first place it was found. Maybe it was in the food, maybe it was in the soil, maybe it was in the water. So really the clinical benefits are more important than the sourcing is the point I'm trying to make. And if you look at the right-hand columns, acid tolerance, most of these strains are very acid resistant, and the main ones we use, which are the LA14 and BLO4, over 90% survive in a simulated stomach acid solution for an hour sitting there, which of course it won't typically sit that long in our bodies. And adherence, the right-hand column, I actually made that from looking at 2 different studies of each one and using the average score on a scale of 4, how well these strains will adhere to intestinal cells. And most of these, they do very well. If you're at 3 or 4, that's the high end. So, these are good strains. They don't need, because they're high acid tolerance, they don't need enteric coating. A lot of companies will enteric coat, but that's really something that was known as yogurt cultures. Thermophilus and Bulgaricus have lower levels of passing stomach acid intact. Survival rate is lower on the traditional yogurt cultures, but when you add acidophilus bifidus, it goes way up. You don't really need special coatings. So the strain needs to be able to resist stomach acid. It has to actually resist a lot of the processing, including shipping. And we do shipping tests on our products. And some of the shipping tests and stability testing we've done on probiotics include keeping it at a temperature of 50 degrees Celsius, which is about 122 degrees Fahrenheit, for 48 hours to see if it will still meet label claim. Because these strains are much more robust when they're dry, and it's when they get moisture and they are activated, they're woken up from their sleep state, that's when they become more sensitive to heat, where say a high fever might kill them in the body. In the bottle, it won't. So isn't that interesting? So the only way to know the potential benefits of probiotics is by looking at the test tube in vitro studies, looking at the live studies in vivo, and looking at human clinical trials especially, because that's really where the claims should be coming from. So when we're looking at clinically relevant strains, I list a few here and some of the products that we use them in. And you can see the top 2 are both the same genus and species, Bifidobacterium lactis. Why do we use BLO4 in some products and HNO19 in others? And the reason is different evidence base. HNO19, you might have noticed on the earlier chart, is a dairy strain. It was actually the top strain identified by the New Zealand Dairy Research Council. And they looked at thousands of strains in their library of strains and determined this was the one that had the best evidence of potential human benefits for health. It's been the subject of a number of clinical studies. It's top rated. It's been even in review studies. And it tends to help with reducing gas, bloating, and GI transit time, which are benefits beyond the general, say, vague immunity or helping you absorb nutrients or those kind of things that might be appropriate for generic strains, but it has some specific strains. That doesn't mean BLO4 has no value. It certainly has its own studies on immunity and other things, but not the gas, bloating, transit time, and being used for infants and throughout the life cycle, including pregnant women. So sometimes the amount of studies and data are much greater on a certain strain. These are name brand strains. They're trademarked. They're restricted who can purchase them and use them. And sometimes if you want more claims, you have to spend more money. There might be a difference in stability and how much overages we have to use to meet label claim through the best by date is another consideration. Shelf stability, there's a lot of factors we have to look at in formulating. And in fact, the HN019 and BLO4 are both in the clinical GI probiotic. We're actually using 2 strains of Bifidobacterium lactis to round out and enhance the benefits of that formula. So, for example, our probiotic 10 has 10 strains of probiotics. Many of them have been clinically validated in studies. The predominant ones are the first 2, the acidophilus LA14 and the bifidus BLO4, but there are still significant amounts of all these other strains to support them. And if you want to look up these strains on PubMed or some other research source, you can certainly see that they've had studies on them to show that they have potential benefits in human health, supporting GI health, immunity, or other issues. And they tend to have not only the acid tolerance, but excellent adhesion to the cell lines. I mentioned that in the columns in the earlier charts. But look at this one, over 90% survival in HCl and pepsin at an hour at a pH of 3 at body temperature. And that's a pretty good simulation of what happens in our bodies. Now, in terms of GMP certification, as I mentioned earlier, Underwriters Laboratory has been our certifier and auditor for GMPs. We're also audited by the FDA. We're also audited by large chains of retailers that utilize us as a contract manufacturer. So some of the brands, if I mention them, you would all recognize, actually use us to produce products for them. And we're typically top rated as a contract manufacturer. We're also top rated by the FDA in terms of not getting 483 observations, which are defined as being in violation of GMPs and getting a notice. So say if you're a retailer or a restaurant and you have a scoring by a health department, imagine getting the top score every time. Maybe it's 100, maybe it's an A. It varies in different parts of the country. Now, allergen control is another area that we think is very important to our consumers. We're a peanut-free processing facility and we only purchase from peanut-free vendors. That tends to be the most dangerous, most deadly allergen statistically. So we do try to avoid having that risk in our products. The other 7 major allergens and other potential allergens we do process in our plants. So we have cross-contamination prevention procedures. We identify allergens coming into the plant. We store them in separate areas of the plant where they can't spill on each other and cross-contaminate. We have airflow and water control and thorough cleaning of equipment between batches to avoid cross-contamination. The FARP system, which is a swab testing of equipment after cleaning to make sure that allergens are no longer present. And we also do air and water testing in the facility, looking for any signs of bacterial contamination that might be present or even moving or spreading throughout the facility. This allows us to monitor potential cross-contamination air ducts and water supplies and drains to make sure that our products are going to be as safe and non-reactive as possible. And by the way, we also use the riboprinter, not only for probiotic testing, but to identify these organisms that we find within the plant. That's actually something the Centers for Disease Control uses as well for monitoring, say, foodborne illnesses or things like that. They actually track it to the source genetically using the same instrument we use. So we are one of the top labs in the world. We're performing over 16,000 tests a month in our labs, microbiology, physical attributes, minerals and metals, fatty acids and similar compounds, botanical identification, vitamin identification, and quantification. We test for contaminants, and we do prep to appropriately be able to test each material. In our liquid chromatography analytical laboratory, we have close to couple dozen of the high performance liquid chromatography instruments, which allow us to do very accurate measurement of identity and quantity in materials. For example, we would use this to measure vitamin C or beta carotene, for example, and be able to tell you, is it in there and how much? But beyond the usual, even though we have a large number of these and probably a lot more than most companies, we have some unusual capabilities I'd like to point out. So when you're looking at this alphabet soup of different instruments, GC stands for gas chromatography, MS stands for mass spectrometer, and we have gas chromatographs by themselves. We have them hooked up and daisy chained to a mass spectrometer or even two mass spectrometers. And what that allows us to do is to distinguish and magnify differences between materials and making the instruments more and more sensitive is really what's happening. So on the next line, the liquid chromatography with one or two mass spectrometers, but maybe a mass spectrometer and a time of flight column. You're actually bouncing on a kind of a trampoline in a vacuum or near vacuum. You're bouncing the molecules and measuring the transit time so that very slight differences between similar things magnify, and you can identify things that otherwise might look identical or almost identical. High performance thin layer chromatography, I'll actually talk about in a minute. IC is testing for minerals and metals. And there's actually different instruments for the metals, which are at parts per billion, and the minerals, which might be at milligram or even gram amounts. When we're looking at parts per billion, we're looking at nanogram amounts. So, there's different scales that require different instruments. We even have a very unusual capability that even most testing labs in the United States don't have, which is being able to distinguish organic versus inorganic arsenic, because inorganic arsenic is much more toxic. And all the toxicology studies actually determine arsenic toxicity based on the inorganic fraction of total arsenic. Yet today, most labs are putting out reports on total arsenic, leaving us to guess at whether it's toxic or not, or how toxic. So these are some of the examples I'd like to point out. Now, I did mention the high performance thin layer chromatography, and I like to distinguish this versus the testing for genetics, the DNA testing. DNA testing is great for crude herbs that are not processed, and other botanicals. But it has limitations. It tells you if it's some of that thing from a certain species in there. It won't tell you how pure it is. It won't tell you if there's something else mixed in there. It won't tell you if there's any actives present or how much, whether it's going to actually be an effective product. And it will not work on extracts, because extracts might not extract the genetic material might actually degrade or break down or give you fragments of genetic material to give you false positives, which has actually happened. And so there's some really serious limitations on DNA testing, and especially on the botanical extracts, it might be actually fairly worthless. Now, thin layer chromatography overcomes those issues. It's the best test for identity. It will tell you if it's the right genus, species, even part of the plant, because we have validated standards to test against. It's the main chemical test for identity in the pharmacopoeial monographs like the US Pharmacopoeia, the USP, or the US Herbal Pharmacopoeia. It's the only test allowing you to completely analyze a sample. And so this is the instrument used for strength, purity, stability studies, not DNA testing. So, DNA testing has a value, but a very limited value. This has actually more value to us for botanical identification, quantification, and even knowing the part of the plant used and the actives. So we're testing a wide variety of materials in different ways. We're testing for identity, we're testing for strength, which is often called potency. Purity, we're looking for adulterants which are deliberately added, whether it's irradiated for sterilization, whether it's spiked or adulterated with pharmaceuticals or synthetics or things that should not be in there. And we also do stability and freshness testing to validate our packaging and our inserts and our dates, our best by dates. Now on the right is contamination. These are environmental contaminants, accidental contaminants. So we're testing for heavy metals, PCBs or industrial chemicals, pesticides, agricultural chemicals, microbiological, and radiation from the environment. This is an example of our metals testing. I'm told that installing one of these instruments is about $500,000, and I believe we have 3 of these that we're using to routinely test all raw materials and all blended mixes to make sure that they meet our standards for heavy metal limits. If we have something like the borage oil I showed early in this presentation that we claim is expeller pressed hexane free, we do have a way to test for residual solvent testing. There are some materials that are solvent extracted, and if they are, we want to make sure we can test for residual solvents. If they are not supposed to be solvent extracted, we can still test for residual solvents to make sure that we're getting the material that we asked for and actually contracted for. So we do have a specialized instrument to look for solvents as well as fatty acids. I mentioned the time of flight equipment to do some very sensitive differences, slight differences between materials. This is that instrument. Very unusual to find this outside of a research lab. Or this one's probably even more rare. It's hand built at the University of Glasgow, and at the time we purchased it a number of years ago, there was a 6-month waiting list for it to be hand assembled at the Scotland University. And it's a way to test for products that have been exposed to irradiation. By the way, those materials tend to have a crystalline structure to hold a radiation signature. Things like oils that don't have a crystalline structure, say fish oil or something, would not typically show any evidence of being exposed to radiation or containing any residual radiation signature because it doesn't have the right structure to actually hold on to that. And for pesticide testing, we probably have the best pesticide testing in the world. And I'm not exaggerating, I'll prove that on the next slide. But this is advanced triple quadrupole gas chromatograph, mass spectrometer, daisy chained together for a liquid chromatography mass spectrometry. And it's an estimated $2 million investment setting up this lab and this instruments, and very high-end forensic and other high-end chemists working for us who know how to utilize this, including method development scientists. And this is something that gives us an ability to do almost a 3D analysis of chemicals to identify pesticides. Uh, at this time, we're looking at something like 600 plus pesticides we can identify, which is about double the USP test for pesticides, is my understanding, and at much more sensitive levels than typically is available. And as evidence of that, last year in August in Toronto, there was an international pesticide conference from the AOAC, which is the Analytical Chemist organization that is very reputable. How reputable? The FDA asked them to set standards for the industry in the country. They set official methods. They set methods that are accepted by the USP. Health Canada, the equivalent of the FDA in Canada, also asks AOAC to set standards and relies on them for testing standards as an independent analytical chemist organization with its own peer-reviewed journal. This is very authoritative, very high-end organization. At their conference last August, scientists from now presented our own methods of finding ultra low level of pesticides in botanicals, despite the interference of the matrix of the pesticides. Components in the pesticides and the botanicals sometimes are so similar that the botanical can mask or hide the pesticide. You can't find the pesticide even if it's there. We are developing methods to find the needle in the haystack, to look beyond that haze. And on the right, you'll see another method for finding glyphosate, which is Roundup and similar type pesticides, in botanicals as well. So finding ultra low levels and different methods of doing that. This slide is a summary of our method. So when I say we have some of the best pesticide testing in the world, this is serious, yes. We've been testing for a couple years an in-house pesticide testing program looking for ultra low levels. And it's the fats, the proteins, the pigments, and other things in the matrix of the plant material, the botanical, that can hide the pesticides and are difficult to recover in a screening method. We're actually helping to develop the methods that allow us to find these pesticides, even at very low levels. We've also demonstrated our methods for determining natural and synthetic amino acids, natural and synthetic substances in like SARMs, which is a controversial type of material now. It's kind of like steroids or androstenedione type materials, and we're trying to screen our materials so that they don't have this in there, an added pharmaceutical. How do we distinguish the IGF-1, the growth factor from deer antler velvet extract, from a synthetic that might be spiked in there? We actually developed a method for that. And the AOAC's first official method, developed at the specific request of the FDA. The FDA actually asked this organization to come up with a standard method. We volunteered to help do that. Our method was chosen as the official method. It was validated by 12 labs around the world that were given blinded samples of our published method to see if they could all replicate the testing and come up with the same results. Is this a reproducible method that any lab could do and get the same results? And because we had such a good method, it became the first AOAC official method. We also published AOAC methods for other things, including SAMe. And in 2011, we published a method we developed to find adulterants in raw materials. These are the Viagra and erectile dysfunction drugs, the melamine and proteins that was a problem poisoning people when they're trying to fake how much protein's in things by using nitrogen-based compounds like melamine. We're looking for diet drugs, looking for different pharmaceuticals. And our method has been validated now by 2 additional research groups who have published follow-ups to our method, using our method and building on our method. And our method is called fundamental in identifying pharmaceutically identified raw materials. This slide is actually a little old. The numbers are actually higher now, but we're producing tens of millions of supplements. We're doing billions of capsules. We're probably approaching 1 billion tablets a year. We've got close to 1,000 SKUs, stock keeping units of supplements, producing a lot of things. And soft gels, we've actually increased our production as well. So we've come a long way. We are considered the largest independent manufacturer in the natural products industry. And we make about 1,500 SKUs in different categories. This is a very small sampling to show you what some of the labels look like. And if you have any technical questions, we do have a toll-free number as well as an email, as well as a form. You could contact us on our webpage, nowfoods.com. And feel free to contact us. We're answering about 97% of all incoming calls live. We're typically here from 8 till 5 weekdays Central Time because we're in Illinois for this office. There's another office in Guelph, Ontario that has also contact information. If you're in Canada, you can contact our product information specialist there as well. And we can answer questions about product quality and uses, ingredients, allergens, potential cautions and interactions, all those kind of things. But we're still a family-owned company. We have family values. We believe we share values with our retailers and our customers. And I'll leave this slide up here. Some of the things that we like to talk about for our quality, some of the statistics on the left, and some of what is not in Now products. You know, there's a much longer list, as I mentioned, on our webpage, but just to give you a sense that we're very conscious of being as natural as possible, as having effective products that are safe, but trying to avoid things that are overly processed and synthetic. So with that, we're going to turn this over for questions, and I do thank you for your attention.